In 2017, above the International Space Station, samples of bacteria were discovered on board the orbital facility's surface. Whilst the samples revealed a number of different sea plankton and soil microbes, its discovery asked an important question on whether these microbes could pose a problem for astronauts aboard the ISS. In space, the habitat is confined to an ecosystem consisting of microorganisms and humans. To ensure our astronauts are able to safely operate within such an environment, it is therefore important to study microorganisms and the methods of microbial monitoring. Before we take a look at our friends above the ISS, let's provide some context on microbacteria and their space environment. The International Space Station is a habitable artificial satellite in low Earth orbit. It was launched in 1998 for the purpose of astronomical research. Recently, there has been evidence that microbes found on the ISS could possibly harm astronauts whilst in orbit. You might be asking, what are microbes and how could they harm astronauts in a secluded spaceship?
In the Learning Outcome 1 of Lecture 6, it is explained that a microbe is a microscopic organism, which may exist in its single-celled form or a colony of cells. Microbes are found everywhere in life, ranging from fungi, bacteria, archaea, algae, protozoa, and viruses. Microbes can be either prokaryotic or eukaryotic. Unlike the eukaryotic cells, prokaryotic cells lack a nucleus and other membrane-bound organelles. The microbes found on the surfaces of the ISS are predominantly classified as bacteria. These microbes have the potential to affect an astronaut's health, as their virulence, which is the severity of the disease, and pathogenicity, the ability of the microbe to cause disease, can be altered under the influence of microgravity. This means that they might behave or affect humans differently in space, compared to on Earth. Similarly, the immune systems of astronauts can potentially alter and be weakened by the microgravity in space, rendering them susceptible to the effects of these microbes. What makes it more concerning is the fact that most of the microbes found on the surfaces of the ISS cannot be cultured under the standard laboratory conditions on Earth, making it harder to analyze them. However, advanced molecular and culture-independent technologies such as quantitative polymerase chain reaction and sequence by synthesis can help scientists identify these microbes.
Bacteria can easily be identified through culture-dependent techniques, where it is grown on an agar nutrient plate and is isolated. Then it can be characterized by its morphology, which is size, shape, and arrangement, and other properties. However, most of the microbes found on the surfaces of the ISS cannot be cultured under the standard laboratory conditions on Earth. Thus, microbiologists utilize culture-independent techniques such as quantitative polymerase chain reaction, qPCR, and sequence by synthesis, SBS, to identify the microbes found on the surfaces of the ISS. Similar to the traditional PCR method, qPCR amplifies the partial 16S rRNA gene in bacteria to increase the number of copies of our desired sequence. The 16S rRNA gene is used as the standard for classification and identification of microbes because it is present in most microbes and shows proper changes. Unlike the traditional PCR method, qPCR utilizes an assay, such as the fluorescent cybergreen 1, to directly monitor the amplification progress. In a double-stranded DNA, the cybergreen 1 dye vibrantly fluoresces, but displays weak fluorescence when binded to single-stranded DNA. This allows microbiologists to monitor the progression of PCR as the DNA strand goes from single-stranded, so after denaturation, to double-stranded after elongation has taken place, resulting in an increase in fluorescence. qPCR has produced enough copies of the bacterial samples. The samples are then sequenced through a next-generation sequencing technique called sequence by synthesis, or SBS. SBS allows for multiple sequencing reactions to occur at a rapid rate, where the specific base order of the gene is determined. This process involves attaching a primer onto the end of each target gene, then free nucleotides are then added in cycles. Each type of dd-NTP is tagged such as it emits a different wavelength or color when it's complementary binded to the template strand. As each dd-NTP is added, a picture of the fluorescent colors is taken. These pictures are then analyzed by computer programs to configure the base sequence of the strand. The sequence of the gene then can be compared to a microbacteria database to identify which microbes were in the sample.
Primary researchers have identified 133 bacterial isolates within the samples. In the tested samples, the predominant bacterial species were the Staphylococcus aureus, the Pentoa conspicua, and the Pentoa gavinea. Some of the identified microbes are considered to be opportunistic pathogens on Earth, which can negatively affect astronauts with compromised immune systems. Other identified microbes are shown to form biofilm, which is an accumulation of microorganisms. This can prove to be problematic as biofilm is known to promote resistance to antibodies, posing problems for astronauts if they become infected. Moreover, biofilm formation can also decrease infrastructure stability by causing mechanical blockages, reducing heat transfer efficiency, and inducing microbial influence corrosion. By further studying these biofilms, scientists can ensure the health of the astronauts and the structural stability of vehicles when undergoing long-term space missions. It was also found that these bacteria, including the Burkholderia capitia, contain contaminants that were found in the portable water dispenser and the portable water system. These contaminants are resistant against decontamination techniques used to sterilize water, such as antibiotics and UV radiation. The presence of these bacteria contaminants can cause serious respiratory infections in patients who are vulnerable to illness. By further researching these bacteria, microbiologists can research better water decontamination methods to ensure the safety of astronauts. While microbes can be useful, such as gut bacteria that help with digestion, there are also many types of bacteria that can pose serious health and infrastructure problems, such as those previously mentioned. Therefore, the study of the microbiome on the ISS can assist in the creation of a system that can alert the crew when there are changes to the bacterial concentration or diversity. These findings can also help produce possible medicines or vaccines that help protect the health of astronauts against bacterial pathogens when undergoing deep space missions.
To sum it up, the identification of these ISS microbes can open up many future research directions to see how their virulence and pathogenicity change in outer space and possibly affect the health of astronauts. To classify these microbes, culture-independent methods such as qPCR and SBS were used, as most of these microbes are unculturable under standard laboratory conditions. The research revealed that 133 bacterial isolates within the samples, with some requiring further research to ensure the health of astronauts in deep space missions. Thank you for listening to our presentation, and now let's say goodbye to our little astronauts.